todos y a todas. Hoy tenemos la visita del doctor Uri Zaragovi en nuestro departamento instituto. El doctor Zaragovi fue director de postdoc de Pablo Barcelona en Canadá. Entonces, bueno, vamos a aprovechar su visita para que nos dé un seminario y nos cuente, digamos, los avances que él ha tenido en sus líneas de investigación. Él va a estar todo el día en nuestro laboratorio, así que si alguien quiere después acercarse a charlar con él, es bienvenido. Y en forma simultánea vamos a también dar la bienvenida a los miembros de la Asociación de Investigación en Visión y Oftalmología, que hoy también nos acompañan en este seminario. Nosotros somos miembros de la comisión directiva, que hemos aprovechado para hacer este doble evento a través de Zoom. Le voy a dar la palabra a Pablo, que lo va a presentar a Uri. Muchas gracias por acompañarnos.
Y bueno, muy rápido, fue eso la inquietud que surgió de poder traer a Uri a disertar acá y que comentara, y sobre todo para mostrarle toda la gente en Córdoba que trabaja en esas áreas. Así que bueno, simplemente Uri, como podrán escucharlo, es argentino, pero hizo casi toda su carrera, inclusive su carrera de grado afuera. Obtuvo su bachelor en bioquímica y química en la Universidad de Brandeis en Boston. Luego, en un programa de graduación combinada, obtuvo su PhD y MD en la Universidad de Miami, siempre en el área de biología celular, molecular y inmunología. Hizo un postdoc en la Universidad de Pensilvania, en un laboratorio de medicina y en el área de biología estructural. Por eso es que muchas de las áreas donde se concentra en áreas de péptidos y obtención de unidades funcionales pequeñas derivadas de grandes proteínas. Actualmente es full professor, tenure de la Universidad de McGill, donde pertenece al Centro de Cáncer y Departamento de Oncología, al Centro de Medicina Translacional y Terapéutica Experimental, al de Farmacología, al de Química Biológica, al de Neurociencias, y últimamente, que se ha creado desde el 2015, al de Ciencias de la Visión y la Oftalmología. También pertenece a la Red de Investigación de Medicamentos de Quebec, que es la provincia de Montreal, y su laboratorio se encuentra en el Instituto de Investigación de Lady Davis, dentro del Jewish General Hospital, también perteneciente a McGill. Y bueno, como dije, digamos, sus investigaciones se centran en las estructuras de macromoléculas y la participación del ligando receptor principalmente. Posee más de 120 publicaciones científicas altamente citadas y posee 21 patentes, la gran mayoría con licencias, y donde ha desarrollado fármacos exitosos desde un laboratorio académico, y ha fundado algunas empresas relacionadas a eso. Así que nada más, te doy la palabra, Uri, muchísimas gracias. Y su charla se titula ahí Academic Translation, Therapeutic Target and Therapeutic Agents, Use of Neurodegeneration for Cancer. Gracias, Uri.
Gracias por la invitación, es muy lindo estar acá. Saben que hablo español, pero el seminario lo voy a dar en inglés. Y quiero comentar que es la primera vez en mi vida donde están tomando mate en la audiencia. Que nadie llame al oficial de seguridad. Voy a tratar de sumarizar en los primeros 3 slides la charla completa, y después el resto de los 80 slides, más o menos, van a ser ejemplos prácticos de cómo hacemos nosotros traslación a la clínica. Okay. Lo que quiero hacer es estimular un poco esa fase que en general los científicos académicos ignoramos o sobreestimamos, o no sabemos hacer o le tenemos miedo. Es posible hacerlo, pero hay que hacerlo bien desde el principio. Y los ejemplos que les voy a dar van a tratar de demostrar eso. ¿Okay? Y lo voy a en nuestro laboratorio ha hecho translación en el área de neurociencia, especialmente en retina, neuronas de la retina y en cáncer. Voy a hablar de tres temas. Voy a empezar en inglés, porque si no, no me sale. So I'll talk about neuroprotective approaches targeting 3 tyrosine kinase receptors. These have actually been translated to the clinic in my lab. More recently, tumor marker gangliosides in cancer have been translated to the clinic. And a very frustrating process of promoting the concept of anti-neurodegeneration as a form of neuroprotection. There's the yin-yang situation. Targeting 2 receptors, P75, and an isoform of one of these pro-survival ones. I think you're all familiar with P75, so I will not spend a lot of time explaining it. So before I start, I need to disclose conflict of interest. So I was a founder of several companies that took the academic work into the clinic. That's not something that we can do because on average it takes $1 billion to get a product to market in the pharmaceutical industry. So if you can give me $1 billion, then I don't need to do it outside. But normally that's how you do it. These companies and other companies licensed a lot of the patents that we developed together with the university in the fields of ALS, neurosensory hearing loss, retinal dystrophies, and cancer therapy. So an overview of the drug development process. This is how it's done in industry, not in academia. So this actually came from, I think it's Pfizer. So they have this in the main lobby as you enter. You start with target identification. You very quickly identify a hit that can bind or modulate the target. And then you go through all the medicinal chemistry. Eventually, somehow you come to the conclusion, this is my lead compound. And you pre-nominate it for clinical development. You do all the in vivo work, ADME toxicity, etc. And then you go through the clinical phases and you launch. This whole process takes on average 10 to 15 years. The shortest one is the discovery process here. And it takes, as I said, about $1 billion plus US dollars to do this in industry. Obviously, there has to be a better way, right? This is a ridiculous process, especially because industry is very, very bad at doing this part. Okay, the discovery process, especially what I call target identification and validation, is very poorly done in industry. They're very, very proficient at doing this part. So this is an opportunity for us. So we're nimble, we're fast, we're cheap, we do things, we're smart, creative, intuitive, and we can do this aspect much better, I think. So, in fact, Goodman and Gilman are each 2 Nobel Prize winners in pharmacology, and they wrote the textbook used in medical school. And they say that in drug, this is part of a longer sentence, but drug development, the process of target characterization and validation are fundamental, but they're understudied or overlooked. So this is an opportunity. And the key is once you find a target through genomics, proteomics, gesomics, which is you read the literature and you figure out, okay, this target may be in my pathway. But then you need to develop a hit to bind to your target and modulate the function of the target. So binding alone is insufficient. It needs to be a functional binding with consequences. And then your target needs to be stable and not disappear or downregulate or desensitize after one shot, one round of treatment, right? So this is process validation. We tend to overestimate. A lot of people think, especially in cancer, overexpression of a particular protein, oh, this is a great target. That's wrong. Okay, the presence of the target doesn't mean anything because the cancer will get rid of that target very, very quickly. In fact, a lot of the immunotherapies, that's what happens. They fail because of that radiation. So you need to, and this is the crux of the problem. You need a hit or a compound to validate the target, and you need a target to validate the compound, right? So it's a vicious cycle. Somehow you have to break it, the vicious cycle. And this is where we come in. So this is how we do things. It's not the only way you can do things, but that's how we do it in my lab. We use protein mimicry as probes and eventually as potential drivers. So mimetics are small synthetic chemicals that mimic the structure of peptides and proteins, the binding properties, so they bind exactly to the same target at a particular hotspot. We define hotspots as the armpit of the target. So if I tickle you here, you react. If I tickle you somewhere else, maybe you don't react, right? So hotspots are functional units of the target. And therefore, the mimetics can mediate biological function either to activate or to be inactivated. So you can evaluate binding, function, and modulation in disease states. So we've been successful, and this is basically, you can stop listening. Okay? So this is the message I want to bring. We've been successful at making monoclonal antibodies and mimetics of them. And this is my preferred pathway. But we made growth factor ligand mimetics and receptor mimetics. And so the mimetics will bind to the same targets as the antibody or to the receptor of the growth factor, and so on. If I have enough time, I'll get to discussing whether mimetics can be designed to mimic non-protein targets. All of these are proteins. Can you make mimetics, for example, against glycolipids? And the answer is yes. So that's why I'm presenting. Otherwise, I wouldn't be presenting. Growth factor mimetic, this is NGF. You probably all know this protein. It's a dimer. So we identified these beta turn structures that are finger-like projections into space. These are the key residues that contact the different receptors for NGF, the growth factor called NGF. And if you make synthetic small molecules that look like this is the synthetic, and this is the crystal, the NMR of the synthetic molecule, and this is the actual crystal structure of the growth factor itself. And they're indistinguishable down to one angstrom or less than that. Okay, so structurally, the mimetics are identical. This is another example. This is an antibody viewed from the business end of the binding site of the antibody called CDR. So on the side view of the CDRs, and this is a mimetic. This compound is actually in the clinic. It was successful at phase 3 clinical trials. So you can move very, very quickly. And I'm showing these as examples. So I'll start with neuroscience and I'll finish with cancer, depending on that. So for neuroscience, we went after this receptor complex, the neurotrophin receptor complex. It's a very difficult one, and that's why we chose it. At the time, there were no pharmaceuticals working on this. So we were alone. There were some academics, but no pharmaceuticals. The receptor complex is made up of the TRAC receptors, tyrosine kinase that homodimerize, and signal particularly through PI3 kinase, AKT, PLC gamma, and ERK. And generally, we tend to think of these as pro-survival. If a neuron has a TRAC receptor and is engaged by the ligand, it will grow, survive, differentiate, make synaptic connections, and maintain the connections. Deficiencies in TRAC lead to neuronal atrophy and eventual death. So it's a great target, in principle. The core receptor is called P75, and I'm sure you're familiar with this one. And we tend to think in a very simplistic way as a pro-death receptor. Okay, especially when it's engaged with it by a particular subset of ligands called pro-neurotransmitters. The problem is, of course, these two receptors interact with each other. They impact on ligand affinity. So you have 9 different ligands, 18 different affinities. It's a mess. It's very difficult. And opposing signals. You have 2 receptors with the same ligand, one saying, okay, survive and make synapses, and the other die or lose or prune the synapses. So we thought, okay, we need to simplify. As complex as I'm saying, this is actually even more complex because the TRAC receptors come in 2 forms. There's an isoform of the protective receptor. We call this the protective on full length with the kinase domain. There's a second isoform that comes from the same mRNA that is spliced. There's a splicing event at the mRNA level that truncates the kinase and has 2 new exons that are otherwise silent. Okay, this receptor acts like P75. It's not P75, but functionally it does the same thing. So again, you have a second level of yin-yang. Okay. What's interesting is that the truncated isoform is hugely upregulated in disease. So you lose the good receptor, and I'm using good figuratively. You lose the good, you gain the bad, and things go bad, right? So you degenerate neurons. The T1 receptor signals, this is work by Esteban and Tesserolo in collaboration. They signal through different pathways because of these 2 new exons through basically G-coupled receptors and RAC. Yet the ectodomain, because it's a splicing event in the intracellular domain, that's the transmembrane, the ectodomains are identical, right? And they both bind the same ligands with the same affinity. Okay, so it's complicated. You have the same ligand binding to 2 isoforms, one killing, one promoting survival, and then the same ligand binding all interacting with each other. Okay, so I think I was drunk when I decided to work on this target. Now, an example of T1 activity is shown here. So the T1 isoform is expressed in retinal glia, in spinal cord glia, in many cell lines, especially in disease, well, in cell lines and in in vivo glia in disease states. And it leads to upregulation of TNF-alpha to toxic levels. So that's one mechanism where T1 is inducing toxicity in 2 neurons. So this is an untreated glia pro-NGF, which is a P75 ligand upregulates both in the biomass and in the media. Sorry, that's NT3. So LPS does it as a positive control, and NT3 does it also, both in the conditioned media and in the biomass. So this is just an example. So in this context, we came up with the following simple concept. We have neurotoxic events and we have neuroprotective mechanisms. In healthy state, you have a homeostatic balance where the TRAC receptors give positive signals, the 75 receptors give negative signals. Again, I'm being very loose about this. In disease state or in injury, there is a toxic imbalance. You fail to protect, you gain activity that is bad for neurons. And on top of that, you gain truncated TRAC receptors that is also injurious. So the strategies are to enhance the positive signals for neuroprotection. For this, you need agonists that activate the full length track, protective, but circumvent the bad receptors. And I told you the ectodomains bind all the ligands equally. So you need to design things for specificity. And then you want to reduce energetic signals by antagonizing either P75 or T1 selectively. But again, how do you antagonize T1 without antagonizing full length? So that's how we apply the mimicry approach. So this is a general concept that we published recently in Frontiers. So when I started in the field, it was the strategy of using the actual neurotrophic factors as drugs. They all failed in the clinic. In Alzheimer's disease, ALS, long list, even glaucoma in the retina, GNF oscillatory therapy, even recently failed. So the problem is that you're binding to both the good receptor and P75, to P75 alone, and of course to T1. So you end up with a net zero gain. So we move on to 2.0, where we made compounds that bound only to track receptors without binding to P75, completely bypassing P75. We made small molecules and antibodies that bound and inhibited P75 itself. And more recently, we made compounds that bound and antagonized only T1 without antagonizing full length. Okay, and I'll walk you through the process of how we did this. And these molecules include antibodies, monoclonal antibodies, and their mimetics, as well as shRNA targeting the two new exons that I told you are added to T1. So we applied all these agents in ophthalmic diseases, dry eye, glaucoma, or I should say high intraocular pressure models. Optic nerve trauma due to battlefield injuries is under clinical development now. This is actually a drug. Is under clinical development. Diabetic retinopathy, wet AMD. In fact, some of the work here came from your own colleagues in a recent paper. I'll show you a lot of data on retinitis pigmentosa, genetically, so monogenic driven injury that leads to photoreceptor death. I'll skip the other diseases though. These are sufficient examples. But all of these are under clinical development in one way or another. So strategy 2.0, pro-survival agonistic antibodies. So we made an antibody, again, by targeting track hotspots, we were able, the armpit of the track receptors, ectodomain, we made antibodies that activated track A, but not the other two, or track B, but not the other two, or track C, but not the other two, to levels comparable to the native growth factor. This is survival assay of neurons, of cells in deprived of serum, so it's standard MTT apoptosis assay. Remember this one, 2B7, because it become critical for the rest of the talk. So in addition to survival, we have endpoints of receptor engagement or biological assays, biochemical assays, phosphatyrosine elevation of the signaling pathways. All of these antibodies bind to each of their own track, but they do not bind or activate P75. Monoclonals are expected not to cross-react in that way. So that was relatively easy. And we end up making mimetics of these receptors. And this is one example that I already talked about. So a track A agonist, we made into a small molecule mimetic that protects cholinergic neurons to even potentiating low levels of NGF So it's a suprapharmacological response. This was successfully tested in models of Alzheimer's, in memory loss, where cholinergic neurons die, so you can protect them, and in glaucoma and other retinal models. And it's now in the clinic. We also made a 2B7 mimetic. This is a TRK-C agonistic antibody. For the non-chemistry audience, the arrows are pointing out the differences between these two. They're not the same molecule. And this is biochemical data showing how these are untreated cells or treated with low or optimal levels of NT3 and the small molecule mimetic inducing tyrosine phosphorylation of the TRK-C receptor. So then we morphed 2.0 into 4.0. So recognizing that these antibodies are activating the full length TRK in a very specific way, the next question is, are we bypassing T1, the toxic form, isoform of the TRK receptors? And that's what we call 4.0. So you want to activate only full length to protect, or inactivate only T1 to prevent toxicity. And it turns out that the 2B7 antibody I told you to remember, unlike NT3, which promotes TNF-alpha to toxic levels, both in the supernatant and the biomass, 2B7 doesn't do that. So 2B7 does not promote TNF-alpha in T1 expressing cells. So it's a full-length selective agonist. How can that be if the ectodomain, the binding site, the primary sequence of the binding sites are identical between the two? I'll get to that. In addition to 2B7, we have, as I said, lentiviruses that silence TRK-CT1 that is being made. We now made selective anti-TRK-CT1 antibodies that are antagonistic and small molecules that are antagonistic to T1, but do not engage or inactivate full length, which is what we want. This is an example of why 2B7 does not activate T1. It simply doesn't bind. So this is a fax where 2B7 is binding to TRK-C, full length expressing cells, but it does not bind to T1 expressing cells. There's no binding, so therefore there's no activation. So this is a Western 2B7 binds to TRK-C, full length, but not to T1, and a T1 antibody binds to T1, but not to full length. Okay, and this immunohistochemistry showing the same thing. And 2B7 activates TRK-C phosphorylation to similar levels as NT3, the natural ligand, but it does it better because it doesn't engage T1, it doesn't engage P75, and it doesn't induce toxicity. So it's a right agonist to use in. We agree? Okay, thank you. So we tested, actually I put the ALS story. So we tested this selective TRK-C full length agonist in ALS. This is the SOD1 for those in the care. The SOD1 model is a mutation that leads to protein aggregation and lack of redox potential in the neurons, so it's toxic and they die. Motor neurons, human and mouse, express high levels of TRK-C, and they're dependent on TRK-C for survival and function. So in this model, the animals get symptoms at 100 days of life. It's a genetic model. It replicates about 5% of the human ALS situations. The others are sporadic. It's called sporadic, we just don't know what's driving. So this animal has the same gene mutation as humans. It develops symptoms of disease, hind leg paralysis at about 105 days of life. And then they are in the controls, they are all dead by 160 days. It's inevitable. We tested with this compound starting at symptoms, so 110 days, give or take, and we gave once a week or twice a week injections, IP, very low dose, 0.25 mg per kilo, which is pretty reasonable. And we stopped therapy here. You can see the survival during the dosing was amazing, 60% and above. When as soon as we stopped therapy, within 10 days, which is the PK of the antibody, everybody died. This was not a planned experiment. This was actually a fluke because we would have continued dosing. We just ran out of compound because we didn't expect this result. So we made enough. So always be hopeful and positive. Yes, it's going to work, make enough, because you don't want to run out. In retrospect, we were very, very lucky because this encouraged us to do it again with continuous dosing and also with much better material. This was material that we made in our lab. The follow-on studies were with GMP material. Good manufacturing practice, no endotoxin, very pure, perfect clinical grade material, and we were able to cut the dose to once a week, 1/4 of the material. So 1/8 of the dosing, and we got better results. So very impressive. And of course, this is survival, but we have rotor-rod performance, body weight, cholinergic phenotype in the spinal cord. This is published, so you can check it out. Fantastic collaboration with Neil Cashman and Bob Rush. I told you we made these T1 specific antibodies, so 2G5, 1E11. These antibodies bind to T1, but they don't bind to full length. In contrast, 2B7 does not bind to T1 and it binds to full length. So these are the antibodies that we're moving forward at the moment. They on their own, they do not induce TNF-alpha. So they're not even partial agonists, but they block NT3-induced TNF-alpha production. So NT3 induces, and the presence of the antibody blocks that induction. So they're potentially really interesting compounds to follow. So how, I told you I was going to talk about the molecular basis for conformational differences and ligand selectivity between full length and T1. So this is a receptor with identical primary sequences, identical, in fact, one mRNA that is spliced and is yielding different conformations in the ectoderm. This is a summary of the explanation. So the T1 isoform has very strong interactions with an enzyme that is present in the ER Golgi called PDI, protein disulfide isomerase. So this enzyme raises the thermodynamic threshold for disulfide formation. So if the enzyme interacts with your target, it will break pre-existing disulfides and rearrange them. So the ectodomain, and there are 8 disulfide bonds in this receptor, the ectodomain of T1 differs from the ectodomain of the full length. And we've shown this by mass spec. So we did limited proteolysis, mass spec, and the non-reducing conditions, and the pairs were very different. And we identified the disulfides that was formed. So it's all PDI enzymatically driven. And we propose that this could apply to many other receptors. See, when you modify the intracellular domain of a receptor, you have to make damn sure that the ectodomain remains the way you like it, or the way that it should be, even if it binds your ligand. This receptor binds the ligand, but it's not the same conformation as this receptor. Okay, and biologically they lead to very, very different consequences. So again, to summarize the in-to-out model of conformation, the T full length has very quick exit sequences within it, within the intracellular domain, and retrieval sequences. These are lost in T1. So it's retained in this path, in this organelle much longer, where it's subject to interactions with PDI. You can immune precipitate PDI with T1, and you can hardly co-precipitate it with the full length. Okay, so it has much more of an opportunity to interact, rearrange, become a conformationally different protein, and now we can make antibodies to it that are selective against one or the other. That was the reason. Interestingly, we prepared what we call delta ICD, so a TRKC receptor that doesn't even have this tiny domain of T1. It's anchored at the membrane, but it has no intracellular domain. It folds exactly like the full length. Why? Because it doesn't have the opportunity to be retained in the area. It flies right through it. So these two are thermodynamically favorable initially, and this one requires the presence of the interaction with PDI. Yeah? Okay. So as just to summarize, we have antibodies, small molecules, vectors, viral vectors. I'll skip this. With high selectivity to one or the other or both, and they all act either as agonists or antagonists. And we use these reagents in vivo as therapeutics. ALS was one example. What's the answer?
Sequencing.
Okay. I'm going to fly through the ophthalmic uses. Okay. So you'll probably know the models. So genetic, inducible, high pressure. This is high noise. You put a mouse through 3 rock concerts. Okay. 100 decibels, 1 hour. It's not a lot. And they go deaf. In fact, if you go to a concert the next day, people have to yell for you to hear, right? Okay, that's synaptic loss. So the cochlear neurons lose connections, and that's why you need higher decibel to hear at the same level. And it takes about a week to get better. If you go through 3 weeks in a row, like the mice, it becomes permanent. So be careful. I'm not kidding. Especially if you're overfused. So I need to worry about it. And it's just like lymphoma. Okay, so there's a risk factor associated with age. I'll go through only retinitis pigmentosa. Okay, so wild type retina, very little TRKC1, very little TNF. The genetic, just 1 point mutation in the rhodopsin, okay, which mimics human disease. Huge levels of T1 co-localizing with TNF alpha mRNA. If you make a knockout of T1 by simply mutating the splicing site, 1 nucleic acid preventing the splicing event, so they make normal full length, but they cannot make T1. Actually, this is the heterozygote. So they make 50% of the T1. They look normal, even in the RHOP background. And these animals are resistant to neurodegeneration. So this genetic evidence validating the target. So this is, and in disease, you get the downstream signals of T1, including ERK activation, that leads to all the bad things that happen with retinitis pigmentosa, including photoreceptor death. So at 20 days of life, the RHOP mouse has lost 30–40% of the photoreceptors. The heterozygote knockout loses less. This is without any intervention, no therapy. This is just 1 point mutation to prevent T1, 50% of the T1. and even more pronounced at postnatal day 24, and this correlates with the thickness of the photoreceptors in the mouse. And we do this also by using OCT in the animal, so we can actually measure very precisely, non-invasively, the thickness of the retina over time. Very quantitative, again, it's all published. No, that's we can get exactly the same result by using antagonists of T1. So this is the RHOP animal retina with vehicle. You can see the ERK activation, or with the antagonist of T1. You totally inhibit and you suppress or reduce the tunnel positive photoreceptors, indicating reduced cell death in that one. Oh, yeah, thank you. Yeah. There goes an immunohistochemistry person, right? Yeah. So, well, anyway, so there's much less tunnel positive death in the retinas in the treated animals. So we have genetic and pharmacological evidence now. And what's very interesting is that, and this is even, again, an ERK inhibitor does the same thing. You reduce ERK activity, which is TRKCT1 dependent, and you reduce tunnel positive photoreceptors. The same thing happens in glaucoma. This is a high intraocular pressure model. In the normal pressure in the eye, there is very little TRKCT1, huge levels after 14 days. This is at a time point there is only about 7% of RGCs dying in this model. There's almost no death yet, and look at the signal. And it co-localizes with TNF surrounding the RGCs. These white balls are the cell bodies of the neurons that are meant to die or destined to die. In the knockout mouse, you have, if you have the same levels of glaucoma, of pressure, is much less T1 and much less TNF-alpha. So genetic evidence, pharmacological evidence, the compounds also protect. These are RGC counts, and this is the TNF-alpha levels that are produced. These are TNF-alpha levels produced after treatment with the compound. So we reduce TNF-alpha, but we don't reduce another cytokine that we follow called alpha-2-microglobulin, and also something that many of you work here. I told you about the hearing loss, so I want pressure, just don't go to rock concert. So I showed you in vivo pharmacological data for these three strategies. We published a lot of the P75, the 3.01. I'll skip through this also, I apologize. We can discuss it one-to-one later. But we made and this is actually Pablo Barcelona published this, so you can discuss it with him. He's a better source. But we tested in diabetic retinopathy, just to give you the bottom line. So in the diabetic model with 47 days of diabetes, high glucose levels, we intervened with ongoing disease at around day 12 and day 26. And you can see the protection of the layers that would have died. So that's untreated, normal, non-diabetic, diabetic, and with very little intervention, we can protect that layer of neurons, and we protect function. So anyway, he can walk you through this himself. So the bottom line is P75 is also disease modifying target in diabetic retinopathy, in glaucoma, and in other diseases. So I just walk you through the way we do target identification and validation. So we made a compound to validate, and we validate, it goes back and forth. So there's a lot of chemistry I didn't go into. The compounds need to appear, we had to make them and design them. I gave you a little bit of an idea on how to design them out of monoclonal antibodies. That's really our favorite approach. Free to ask questions, by the way. Don't wait until the end.
Tengo una.
What?
I have a question with the
Okay.
with P75 because we published many years ago that the P75 is actually produce pro-survival in motor neurons after lesion, and you have the P75 paradoxic increased expression in ALS patients and ALS models. And it's not clear to me how do you select your antibodies in terms of the molecular complex at the cell membrane of P75 or either pro-NTF. So like, for instance, the antibody is an IgG, so I assume that it will bind, with two arms to receptors.
Yeah.
I don't know how if the antibody combines the trimer that forms, for instance, P75 or not, or just, how is really acting, because you are just talking about competing with the natural ligand, but at the same time you can disrupt natural organization of the cell membrane. And in the case of P75, you can have a pro-survival effect under P75 activation, at least in our hands, but it's not the classical primeric form of the receptor, it's an monomeric form.
Yeah, okay, so it's a long, complicated question.
Yeah.
It's a very good question. It's a very good, but it has many parts. So first of all, I'm aware of the pro-survival reports on P75. That's very cell and tissue and disease context dependent, right? So P75 will promote survival and migration of melanoma. So it's a pro-cancer effect, but in neurons, I think 99.9% of the field will say that it's pro-death, pro-pruning, unless you're studying embryonic development. So in embryonic development, most of us are born with more neurons than need, and we screen them. We get rid of the ones that are non-functional, and it's a P75 dependent mechanism. So everything's cell context dependent. So I'll just say that the pro-survival function of P75 has never been demonstrated in. It's only been done in culture systems. Let's start with that, to my knowledge. With regards to dimer, trimer, monomer, it's a great question. We still don't know. There is a big debate in the field as to which complex does the killing and which complex does what. Our small molecule that I just showed inhibits both ligand-dependent and ligand-independent functions. So P75 has ligand-independent actions also. So it's not just death and ICD is not just promoted by ligand. So we got lucky in designing a compound that block both functions, and we're trying to now at the moment. In fact, we're looking for a collaboration here to determine which isoform we're inhibiting the most, not isoform, which complex, dimer, trimer, tetramer, or ICD, we're inhibiting the most, and that what the in vivo consequences are in the retina, for example, right? So these are very difficult studies because it's been very hard to find these complexes in vivo, right? So a lot of the work's done on cell lines, and you can do anything you want with cell lines. Doesn't mean that it's wrong, but it can be misleading you into, So we recently did a lot of work on dimers, trimers for melanoma. There is no mono in melanoma for P75. You don't exist. It's just not an entity. Is that why it's pro-survival and metastasis? So anyway, it's a long question, a long answer. I didn't give you an answer. Just say I disagree with the pro-survival function in vivo, and we're working on understanding what the complexes are doing.
I have a question.
Mm.
In addition to OCT, have you evaluated ERG or visual acuity?
ERG, yeah. So we do ERGs in the retina, and we do what's called ABRs in the hearing system. So auditory brain stem recording. So it's the same. It's an ERG of the ear, and it tells you how much your brain can hear. Your brain can actually hear how much signal gets to there. The same as the ERG. Okay, so yeah, I want to give credit to the people who did some of this neuroscience work. So obviously, where is he? So Pablo did a lot of it, and other members of the lab that he worked with. The ALS was in collaboration with Bob Rush in Australia, Neil Cashman in Vancouver. Mike is a colleague in Montreal, great collaborator also, expert in neovascularization. And a lot of Spaniards, Enrique de la Rosa Cuenca, Jose Nicolás, and Jose Maria Frade in Madrid. So next, I want to get here. So can we make mimetics that bind or do things behind their sides? And the answer is yes. So as a quick introduction, gangliosides are glycosphingolipids with sialic acid. They're not proteins. They have it's a two lipid tails that traverse one of the layers of the bilayer of the plasma membrane, ceramide head, and 3 to 5 to 6 sugars. So they are, you can think of them as 4 or 5 amino acids, that they're that small. Okay. Yet they regulate all kinds of biological processes because they raft, and they either allow the inclusion or they exclude certain proteins from rafts. So they are signaling by regulating raft formation and raft complexes. So both myself and Jeff Friesco and many others, in fact, the most recent Nobel Prize winner, Carlo Bertozzi, demonstrated that these carbohydrates generally regulate fluidity, raft, and they can signal. So you can get, for example, direct ligand independent activations of any receptor tyrosine kinase that you can think of. So by lowering the threshold of receptor dimerization and self-activation. So it's an oncogene, but it's not a gene and it's not a protein. Very importantly, what she showed is that these sugars are all immunosuppressive. So they both locally and systemically, and we call them the glycocode or immune glycocheckpoint. So you can think of them as PD1 or PD1F or TIM3 or any of the conventional ICI proteins that prevent immune reaction against cancer. We are focused on a number of these gangliosides that are very specifically called tumor marker gangliosides. There are normal gangliosides like GM1 pretty much in every cell of your body, but slight modifications, and the biosynthetic pathways are complex, can make these not only oncogenic or tumorogenic, but they are markers of cancer. So these are present in embryonic development at very high levels, but in the adult they're very, very low, in only very few cells. So we focused on GD2 and GD3. It turns out that they're very highly expressed in melanoma and in ovarian cancer. And so this is old literature. Actually, you can go through exactly 50 years or 55 years of literature where they're described as tumor markers, yet they're not in the clinic. So there has to be a reason, right? Why they exploit it. So the reason of these 50-year failures is that it's very difficult to make antibodies to carbohydrates because they're T-cell independent. They are themselves immunosuppressive, as I mentioned. So even after 55 years, there's only like 4 antibodies to these targets worldwide. Okay, it's telling you how difficult it is. And vaccines fail, all clinical trials were discontinued. So you can conjugate G2 to KLH or any kind of carrier, inject, and you get it's bad news. It didn't just fail, it actually caused a nauseous effect in humans. The concept is that they're purely immunogenic, so a tumor marker is not the same as a tumor antigen, and that's not the same as an immunogen. If you want to raise monoclonal antibodies, you need a really good immunogen that gives you selective responses. So that's how we approach it, using a mini-3 approach. And we publish this in these 2 papers, one in 2012 and one in 2019. So first we recognize that the sugar, the carbohydrate portion that is in the ectodomain changes when it's in the free state or the bound state. The bound, so you can see that this alic acid is actually flipping over. And this is the immune recognizable state. Okay, so when it's floating, it's not going to be very easily presented. But so we made a mimetic synthetic antigen that looks like this. It's a fixed tertiary structure in water, in space. Okay, that was the first finding. We also replaced, this is synthetic carbohydrate, and we replaced it's not shown here, but it's a ceramide and lipid tails. We replaced that with phenyl group to mimic the ceramide. This was kind of a naive thing to do, but it worked. And this was a much superior image. So we were able, in one campaign, one monoclonal antibody campaign, we made 11 unique monoclonals to GD2. Whereas in 55 years of big groups working on this, there's only 4. And of course, we did the same thing for GD3. So these immunogens are therapeutic. This is a very impressive figure I made the cover of Cell. This is the tumors that go to the lung, these black dots, and treated with a vaccine. We call it very loosely vaccine. It's not a vaccine, but it's an immunogen that generates all immunity to the target, and it gets rid of the tumor. Now, if you let these animals go, this is after 15 days. If you let these animals go longer to 23 days, that's how black the lungs become. And these are non-smoking mice. Okay. Yet, when you treat them, you see a very significant reduction in metastasis, in tumor burden. So we made this synthetic antigen. We replaced the problematic portion with a synthetic aryl group that then you can conjugate to anything. We were very successful using BAMAM, just an inert carrier. You buy this anywhere. And then after immunization, we can show zero reactivity to G2 expressing cells, G3 expressing cells, but not to controls. And there is some antibodies that cross-react, they bind to both. Because, of course, the only difference is this one little sugar. So if you're binding to this side of the epitope, again, it's a tiny epitope, right? Yet there is some antibodies that bind to both G2 and G3, some that only bind G2 or only bind G3. And we use these antibodies in immunohistochemistry for ovarian cancer. We can detect 99% of fallopian tubes with tumors or ovarian cancer tumors. of all stages, stage 1, 2, 3, and 4. This is unique. There's no literature anywhere showing this for ovarian cancer. In fact, ovarian cancer is called a silent killer because there's no diagnosis. So women present usually very late disease. Doctor, I have a problem, I have some belly pain, oh, it's gas, go home. Or it's your period, go home. Take an aspirin, come back in 3 months. So what happens is from patient presenting to an expert, I don't mean a general practitioner, I mean an expert. So an actual true genuine diagnosis is 9 months to 12 months. And that's sufficient for a stage 1 to go to stage 3 when it's incurable. So it's a terrible situation for patients and their families. So based on this, we were encouraged to develop a liquid biopsy as a blood test to detect the presence of G2 and G3 in circulation in small amounts of serum. Okay, and we just published, I'll go very quick. So we just published this in Frontiers. We developed an ELISA for ovarian cancer from small amounts of blood, so you can do this longitudinally over time. You don't need a fallopian tube or an ovary. You don't need to go to surgery. And we can detect 98.7% sensitivity and specificity for all forms. And we've done over 700 patients by now. This is compared, including stages 1 and 2, where you absolutely need that diagnostic, so you can treat them early. And we've compared this to the standard of care today in the clinic, CA-125 and HE4, and it's a big p-value. That's actually it's a very small p-value. Very significant differences to the current standard of care. We just started as a priority clinical trial enrollment for an FDA approval. And so we're super proud of this work because the speed, we started this about this basic science about 14 years ago, but the clinical work, we started March 2020. And you all know what happened that month, right? COVID. So we went through COVID and we did all this in spite of all the challenges. So it can be done. And this is one very impressive example. So that's for all stages. The green and orange is our test. And this is the best situation in the best gynecological oncological hospital in the world. Okay, the best they can find is 70% or under that. We pick up nearly 100%. Now, if you look at the patients that are failed, stages 1 and 2 that are never picked up, only about 20% of early stage patients are picked up by the current tests, CA-125 and others. That's in the best conditions if you go to the best place in the world, and this is our test run in my lab, which is not the best place in the world. And it's a cheap ELISA. Compared to the current test, HE4 costs $990 US dollars per test, and it's useless. And we think that we can save in North America alone 50,000 lives a year and reduce unnecessary surgeries that remove at least one ovary and one fallopian tube by 20,000. And they are necessary because they're suspected of ovarian cancer, but until you take out the ovary, you don't. Okay. And we're developing additional methods. I'd love to collaborate with people here, for pan cancer tests. So if we start expanding to the other two more ganglioside markers, we can start bringing in other differentiating one tumor from the other, use different targets. So, and we just had yesterday issued a patent on this pan cancer methodologies using capturing extracellular vesicle. obviously, we're start using the antibodies also therapeutically, as it's called passive immunity, standard clinical development. So this is the best anti-PD-1 in this model. That's the response. That's our anti-G2 antibody, and that's a combination. And this is a Kaplan-Meier survival curve. It's looking very promising, and we're preparing the manuscript at the moment. Yeah, so just to summarize this part, I hope I didn't go too much over time. So G2 and G3 are very good validated biomarkers and targets, both for diagnostic and for therapy in melanoma and ovarian cancer. We picked one winner of the 11. We think clone 4 is the one, but there could be the other 10 following closely behind. And I want to thank the people who did the work on this aspect. Ali is a fantastic postdoc, together with my team, and it's in collaboration with colleagues both local to my institute and nearby and elsewhere. In particular, the structural work that led to the synthesis of immunogen was done by Wen-Yong Tong, also a fantastic postdoc who's now director of one of the top 2 pharmas, senior director. So he's done very, very well. And Alba Galan, who worked in 5 papers with us. So yeah, it's a great team, and I'm very proud of their contributions. And remember this. So you can make mimetics, you can use them, you can make mimetics to known protein targets, and you can make it to the clinic. But it takes a lot of energy, that's for sure. Thank you.
Bueno, muchas gracias, Uri. ¿Preguntas? Vamos a empezar con el auditorio y después vamos a ver si hay preguntas en Zoom.
I have a question with the initiation protocol, because I really don't get exactly how was the definition of the conformer for G3, for instance, because I'm thinking that those G3 are not usually expressed on those cells, and you have overexpression, high density, and selectivity of the conformer expression for G3 and these. And it wasn't clear to me how you pick up the monoclonal cell. My strategy was, my strategy was to produce those monoclonal antibodies, for instance, in mice, using animals known for gangliosides, and then you can have like a really, you really increase the amount of clones you can select, but I'm not quite sure
the screening was done against native gangliosides, either on the cell membrane or on the plastic dish. That it was just a standard binding assay. I think the advantage was that the immunogen is the bound state conformer. It's an induced fit. I didn't say that. So the free state is floppy. The bound state is an induced fit. So the ligand makes it that way, and it's immunologically visible conformer. So when you're doing it, regardless of whether the animal is null for the ganglioside, so you increase your chances. It still needs to see it. And when I don't know what you're injecting, tumors or what are you injecting?
No, I mean, we have a passive immunization model for neuropathy, autoimmune neuropathy. That's why.
Okay, so it's experimental. Okay.
Yeah.
Okay. Anyway, we should discuss. I mean, I'd love to use the null mice without antigen. I think it's just going to give 90.
And an old concept about anti-ganglioside antibodies that was used for GM3 at Comoris lab was the density-dependent binding. So yes. I was wondering whether you measure by SPR or similar technology the affinity of the antibody.
Yeah, we
to
do.
determine whether you need identity of the target in order to bind it.
So we do SPR, where you mobilize the target on a chip. Within the different densities, we use standard, methodologies. So I cannot answer that. We know the affinity, but at the end of the day, it doesn't matter because tumors express very millions of GD2 or GD3 per cell, millions, compared to sensory neurons, which is where the neuropathic pain comes from. Neurons may have maybe at base 10,000 versus millions. So in the end, we don't really care. Now, if you were to make a density-specific monoclonal, then you could bypass neuropathy, right? Maybe that's what you're thinking. So you can treat tumors without cutting.
Well, that would happen with the anti-G2 monoclonal antibody.
Well, our antibody, I flew through it, but our antibodies don't cause pain. We tested it.
Yeah,
Both in vivo and in vitro. So this, I told you G2 or G3, two things on the membrane. So one of the things that an antibody to G2 does is to activate an NDA receptor channels. So you get activation of pain pathways that is not blocked by morphine because it's direct intracellular activation. And in fact, in patients that receive this one antibody that we're talking about, you get morphine intractable pain, so high-grade pain that cannot be blocked by morphine, comparable to childbirth pain. So some of you know what that is, right? Very, very powerful abdominal pain. Ours don't do that. We don't know why, and maybe they're density dependent. Maybe we just don't know why, but that's what we picked for. It's the one that doesn't cause pain.
Ruben?
Ruben, thank you for your very inspiring lecture. I would like to ask you how you deal with the pressure in academics to publish and to patent. What's your strategy or advice? Because sometimes we are afraid of getting into that field.
Yeah, so discussing the fear. So don't fear your colleagues, okay? I mean, that was actually my first battle. When I started patenting things, everybody came after me. I just gave him my thing. Okay. I mean, I believed in what I was doing, and so you can criticize me, and in the end, I was proven right. So don't be afraid. I mean, if you believe in your science, you can do both. So you can file a patent, provided you get support for that, and the paper at the same time. So and then the paper is going to take a lot longer than the patent, right? Because of reviewer 3, right? Or reviewer C, that always gives you a hard time. So by the time the paper becomes public, the patent is already well on the way. So you have a priority date, but it was interesting at the beginning. I had to fight my local battle and get the financing from the university. Luckily, McGill had a tiny small budget, so they did give me support. But after 3 or 4, the university agreed to give me all the rights, which means that I had to pay for it myself, which I did. And I did the right thing because the licenses paid back. Right? So it's a risk, but crossing the street, it's a risk. Yeah, if you don't do it, one thing I learned is if you don't do it, nobody's going to do it for you. So the idea that, somebody in the industry will recognize the high quality of the work and the importance, forget it. They won't. You need to do it yourself, bring it to a valuation point where now they become interested. But that, we call it the value of them, is you have to craft it on your own. And if you have a really good colleague to do it with is easier, it's better, because you cry to them. In my experience, the only way that it works is not just any colleague, somebody that you truly, you can have a beer and you can have a fight with and then kiss and make up. Yeah, if not, it's the well is poison. You can have a best science, a best colleague, but if you don't have that element, it's like a marriage. If you don't have that element, it's gonna fail. Guaranteed. The first challenge, you're gonna break up.
Mario?
Hi, Yuri. How are you? Very nice presentation. Going back to the first part about drug activation, you mentioned small molecules. Yes.
Yes.
I remember there is a couple of papers in the United States about a derivative of acetyl serotonin working very well on the
Attractive.
attractive. Have you tried any compounds like that?
Yeah, we did. Yeah, they do work. They don't, the mechanism report, but they don't report the mechanism. They suggest that it's binding to drug. It doesn't bind to drug B. It actually promotes BDNF release. That's the mechanism. So when they make BDNF, it activates drug B. So yeah, we actually are making analogs of those that we can patent. So yeah, we have made
Yes.
a patent with that.
Yes, I have everyone. Many thanks, Yuri, for your nice presentation too. My question is about that you use monoclonal antibodies for treatment. Have you treated also the camelid-derived antibodies? Have the same effect or no?
You mean humanized?
Yes, humanized.
We do humanize. I mean, they all have gone through the process of it.
Yes, but with a smaller antibodies, camelid one that
The camelids.
Yeah.
No, we didn't go through. We have the murine, humanized, and small molecule. So we don't go in between. We did play around with single chain,
Yes.
these which are smallish, monomeric. We had two successful cases. The only reason we did it is we were asking, does it take a dimer or can a monomer activate without inducing dimerization? A monomeric ligand activate without inducing receptor dimerization? And the answer is yes. Actually, we had to fight the field for 2 years. So we sent it to Science, it got rejected because that's against dogma. We sent it to Nature, against dogma. Then we sent it to JBC, rejected. I called the editor. He accepted it 10 days later in full. And 3 months after, there were Science, Nature papers saying monomeric ligands can activate it. Yeah, typical, right? So the single chain FEs we tried and they worked. We didn't go for camelids. Yeah, they were too expensive to make at the time. Now they're cheaper.
Yes, now they are cheaper. In addition, you can produce once identified, you can produce them directly by expression in E. coli, for example.
Yeah. Well, so single chain and yeast hybrids, screening. I mean, all of those are just technologies, right? So you can apply the latest flavor, of the month. But the concept is that this very much doable. I want the students to ask questions.
We are the students.
Curious about how you brought to design a peptide. I don't know if it's a peptide to inhibit drug C.1, I think, and differentiate drug C the full length.
So
The structure.
so the full length and if you want, take, as I showed, different conformations to the disulfide arrangements. So you can actually design immunogens to target them, especially if you know how the disulfides are linked with each other. So that's how we did it. So we made the monoclonals that way, and then we tested for function. Is that your question? Or?
Yeah.
And the small molecules are not peptides; they are really organic,
Okay.
drug-like. I mean, it has better pharmacokinetic properties than peptides.
What about alpha-2 macroglobulin?
What
Have
about-
you tried to modify or
No.
no?
Well, we activate it. we make it,
Okay.
but no, we didn't.
And the receptors? No?
I'm leaving that to Pablo and people like that. But, no, we didn't go after. There is people. I mean, what's his name at Harvard? I mean, he's going after LRP1 in neurodegeneration. Big Alzheimer's guy. Anyway. No. So these people working on targeting LRP1 in AD and things like that. But I didn't. There's really very little. There's one small peptide. What's the name? This is one small peptide that inhibits binding to the RAM. Yeah. So, but
Yes.
at some point we thought about analoging it and making small molecules, but like we haven't.
Okay. Another question?
Question related to alpha-2M, when you show a decrease, that one compound decrease TNF alpha, but not alpha-2M macroglobulin. Do you think maybe this alpha-2M maybe could be toxic effect?
Yes. Yes. Absolutely. I think alpha-2 is a great target, and LRP1 are very good targets. Pablo published one or two papers in my lab on that. That experiment was alpha-2M was a control. So we think that alpha-2M is regulated by P75. Let me rephrase. P75 regulates TNF-alpha and alpha-2M production. TRKCT1 regulates TNF-alpha only. So when we use the TRKCT1 antagonist, we reduce TNF-alpha, but we didn't reduce alpha-2M. So it was actually a specificity control. And it was exactly as expected.
Thank you.
Okay.
Thank you.
Thank you very much.
Facultad de Ciencias Químicas.